region by region. Of course, is a systemic class. You can see that in the table of contents of your book. And so this is our first system. Next thing to say about the integumentary system is that it is a system. People, including myself, tend to think of it as just the skin, but what is the skin? It's an organ. Okay, so the skin is the integumentary system, what the stomach is to the digestive system, or the lungs are to the respiratory system. But when you talk about the integumentary system, it seems like all you talk about is the skin, and so people sort of forget organ versus system. The skin is the largest organ in the body. Straight from exercise 1 or 2 in the lab manual, I don't remember. It's the only organ that's not contained within a cavity. But if the integumentary system is a system and the skin is just an organ, well then what are the other organs of the integumentary system? Well, technically the integumentary system consists of, quote, the skin and its derivatives, unquote. The skin and its derivatives. Why am I getting all these blank stares? Is this 202 instead of 201? You guys are just still asleep? Pants aren't buckled? You were all looking at me like...
We're summing it up for the histology lab practical.
Ah. Histology lab practical is easy. Most common grade will be a B. Of course, if you didn't study, it's not easy. Can't do anything about that. All right, so functions of the integumentary system. One is body temperature. Just reading straight off your notes, you can see them there. Don't have to write them down. Several things go into body temperature in the integumentary system. One is fat. So you think about what you know about the skin just from histology. You have an epidermis, dermis, hypodermis. You know the tissue of all of those. Keratinized, stratified, squamous, dense, irregular, adipose. So body temperature is important. You can think about blood. Of course, where is the blood? Epithelial tissue is avascular. It gets its support both physically and metabolically from the underlying connective tissue. So the blood is in the dermis, but we do shunt blood around. And this is something called vasomotor control. Vaso is vessel, so this is motor control of your blood vessels. If you look at a cross section of an artery, well, you know there's a lot of elastic connective tissue in there, so the vessel can expand and recoil, but there's also smooth muscle tissue in the walls of blood vessels. So blood vessels can constrict and stop the flow of blood to a particular area. Blood vessels can dilate and increase the flow of blood to a particular area. After you've eaten, and this has nothing to do with the integumentary system, but it is vasomotor control, after you've eaten, you send more blood to the abdominal organs so that you can, all the digestive processes. That's why your mom or your grandfather, somebody probably told you, you can't go swimming, you just ate, you'll drown, your legs will cramp up. So you compromise muscle by sending more blood to the abdominal organs, the digestive organs. If you're running, you're going to send more blood to the working muscles. But if it's very cold outside, you're going to constrict the vessels that carry blood near the surface of the body. Because if the blood moves close to the surface of the body, it's 20 degrees out here, the blood's going to become cold. And when that blood is back deep in the body, vital organs will become cold. So to keep your brain, your liver, your lungs, important organs alive, you don't send blood to the superficial parts of the body when it's cold outside. That's where vasomotor control comes in with the integumentary system. It comes in with many other systems, as you'll see over the coming months. On the other hand, if it's very hot outside, you do dilate the vessels and send all of the blood toward the surface of the body. You've all seen Goosebumps. And what does that have to do with cold? Well, really nothing in people. You have a muscle attached to every hair in your body, and we'll look at those in a minute. But every single hair in your body has a muscle attached to it, and you can effectively make those hairs stand up. Now, you and I can't really do that. I mean, you might have seen short hair on somebody's arm kind of stand up, but for the most part, when we undergo this phenomena, which is called widespread pillow erection, a term you'll want to throw out at your next dinner party with the neighbors, widespread pillow erection. That is the widespread contraction of all the muscles attached to all the hairs in your body. But other animals use this to stay warm. So dogs and cats don't just hiss and make their hair stand up to look bigger. They do that to scare away predators. Yeah, they do that. But they also do that to stay warm. Hair is an excellent insulator. Hair works kind of like a goose down jacket. And so the animals that live in the cold environments, like the sled dogs, for example, they use this ability to make their hair stand up to trap air, to remain warm. Under functions, letter B says protection. Well, absolutely, your skin is your first line of defense in keeping microorganisms out of your body. You also produce secretions, glands, that is, produce secretions. And so the pH of your skin is not particularly conducive to microorganisms thriving there. I've got perception listed. Your skin is filled with various receptors for temperature. So this is hot, this is cold, this is sort of in between. Receptors for texture, this feels like sandpaper, this feels like glass, this is sheetrock. You can perceive all sorts of temperatures and textures. You have receptors for pressure. Is it deep pressure? Imagine that you lay your hand on the table and I put this on your hand, you can feel it, and then I add this, and you it's via pressure receptors that you feel the increase in weight, and you could keep adding things and you could perceive that. It's all of these receptors that allow you to do some of the fine things that you do, like thread a needle or start a nut on a bolt. And some of these things you do without even looking. You've done it looking, and you know what it looks like, and you know what it feels like, and now you can reach behind the thing and do it by touch. So there are lots and lots of receptors in your skin. Of course, certain parts of your body, like your lips, have more receptors than others. The tips of your fingers have more receptors than other parts of your body, like, say, just your thigh. I've got excretion listed, and I want you to add there, and secretion. Excretion and secretion. Excretion and secretion are words like effect and affect, take and bring. Nobody really knows the difference between them. Well, you probably all do, but most people don't. If you excrete something, it leaves your body. If you simply secrete something, it leaves a cell or a gland, but it doesn't leave your body. So goblet cells secrete mucin. They don't excrete it. And mucin's not jumping across the room when the goblet cells release it. you excrete oil, you excrete sweat, okay, because it leaves your body. But there are other things that the integumentary system makes that are secreted. I think you probably all know that with the aid of sunlight, your integumentary system can make vitamin D. Well, you don't excrete it. I'm going to squirt you with my vitamin D. Did you feel it? Wipe it off. It's on your forehead. Okay, when you're in the sun, your pigmentation increases. You get a tan. That's melanin. Well, you don't excrete your melanin. It doesn't, you don't wipe it off your forehead or something like sweat. So excrete, secrete. All right, finally, letter E, synthesis. The integumentary system synthesizes a lot of things. Oil. Think about going a week without washing your hair. Don't know why you would do that, but imagine you did. Sweat. You're outside working in the yard, it's hot, you're sweating. That's another aspect of temperature control. But there's a second type of sweat, and we'll talk about these two types of sweat. For now, just appreciate there are two types of sweat. You have sweat as in I'm hot and sweaty, but you have sweat as in body odor. So we've got oil, we've got sweat, we've got milk. Earwax. Keratin is something produced by the integumentary system. You know all about keratin from studying histology. All right, Roman numeral 2, it says skin and overview. Well, the overview is this. Epidermis, dermis, hypodermis, and I'm not going to stand here and tell you what you learned in histology. The epidermis is epithelial tissue, is stratified squamous, keratinized stratified squamous. The dermis is dense irregular, the hypodermis is adipose. The only thing I'm going to add to this is that I've lied to you. The dermis is not 100% dense irregular. It's okay for today and Wednesday when you take the practical. You don't have to change that. But when we get to the dermis, I'll have to tell you the truth about the dermis. So your overview is something you had when you came through the door this morning. Letter A says types of skin. Well, you have two types. You have thick skin with 5 epidermal layers. And you have thin skin, which has only 4 epidermal layers. Now, it's kind of impractical to look at somebody and count the layers of their epidermis, but it's actually very, very easy to make this distinction. Thick skin lacks hair, and thin skin has hair. Now, this is any hair, okay? This doesn't have to be the hair on your head, or you have hair on your face, you have hairy arms. It's any hair. If you go home and you look in the mirror, male or female, you all have hair on your nose, okay? It's really fine. You all have hair on your cheeks, you have hair on your neck, male or female, it's there. You have hair on the, the underside of your arm, places where you don't think you have hair, you do. So thick skin has absolutely no hair. So where is that? Yeah, your palms, your soles, your lips. It's pretty easy to see the two types of skin without counting the layers. All right, letter B, epidermis. You know it's an epithelial tissue. You know it's keratinized stratified squamous. You know it has 4 or 5 layers. The dermis, you know it's a connective tissue. You know it's dense irregular. You know I've lied to you. It's not really, but we'll get there. I think we have an entire Roman numeral on the dermis on the next page. We do. Finally, letter D, the hypodermis or subcutaneous adipose connective tissue. It is your subcutaneous or hypodermis that is important in insulation, so back to the temperature control, is important in padding and protection, determines the shape of your body. Okay, so now let's go through the epidermis. So this is Roman numeral 3. So first of all, just a classic picture of the skin. You can see the upper, oh my God. That is a nasty looking bug. Anyone care if I kill it?
I wanna see.
If you don't want me to kill it, come get it and take it outside, because I'm not scared of bugs, but I don't particularly like them inside where I am. I think they're called camel crickets. I've seen them as big as a frisbee. About killed it with it. You want to take it outside? You go right ahead. 10 extra points on your final exam for saving the bug. All right, so, all right, well, that's so sweet. We'll wait on it. In the interest of your kindness, so histology practical. Name the protein that forms the indicated structures. Come on, somebody blurt it out. All right, so epidermis, dermis, hypodermis. I'm going to zoom in on the epidermis. So this is a photomicrograph from your textbook of the epidermis. We see how the dermis and the epidermis come together like interlocking fingers. I refer to that as the interface between the epidermis and the dermis. The layers of the epidermis are shown over here. So we've got stratum basal, stratum spinosum, stratum granulosum, stratum lucidum, and stratum corneum. With regard to the thick versus thin, the thick having 5 layers, the thin having only 4, it's the stratum lucidum that's absent in thin skin. I think you'll see that indicated on your notes. You do. Roman numeral 3, letter B. Item 4, stratum lucidum. There's an asterisk there because that is the layer missing in thin skin. In anatomy, we use a lot of mnemonics, not really in physiology, but in anatomy, we use a lot of mnemonics to keep up with things. Mnemonics are stupid little rhymes, often filthy and cannot be repeated in public. But you can certainly Google them if you're interested. But there's one right here that people have used since the beginning of time. So you take the first letter of each word. Boys say gorillas like candy. So if you want to use it, use it. If you don't, don't. Boys say gorillas like candy. Stupid but catchy, and hope you keep straight the layers. Okay, regarding the stratum basal, the deepest layer, it's a very thin layer. And it's a layer that's mitotically active. The stratum spinosum, as you can see in the picture, is very thick. It's also mitotically active, not as mitotically active as the basal, but they are both mitotically active. Then we have the really thin granulosum, the very thin lucidum, and the extremely thick corneum. Now the corneum is thicker in this picture because it's thick skin. We know it's thick skin because the lucidum is there. If it was thin skin, there'd be no lucidum. Also, if it was thin skin, we'd likely see a hair. Anyway, back to the mitotic activity of the basal and spinosum, but primarily basal. So there's a constant migration of cells. New cells are being produced deep in the epidermis, primarily the stratum basal. And if a cell down here divides, the daughter cells are displaced toward the superficial aspect of the skin. And so we have this constant migration of cells. Because of the migration and the underlying forces, the cells are flattening. So what starts out as round or square becomes less and less. And as the cells rise, they flatten out, they become squamous. You already know that we name stratified tissue based on the shape toward the free surface. It doesn't matter what shape or shapes we have down deep. So the flattening is because of the underlying forces, this constant migration from the parent cells producing new daughter cells. Also, as the migration is occurring, the protein keratin is being produced. It will be a subject for another day to get into all of that, but as with almost every protein in your body, it starts out as a precursor, it has to be modified, it has to mature. So keratin starts out as something called keratinohyaline, which you do not need to write down, but the keratinohyaline matures and becomes keratin during this migration. And as the keratinohyaline matures and becomes keratin, the keratin does what you already know it does. It dehydrates the cells, they die, they lose their nucleus. And so up here, what we have are just dead keratinized cells. They don't even look like cells. Got a couple of pictures in this folder that you have in your histology slide set. So you've all seen these, and this of course is the epidermis, this is the dermis, little sub-Q down here. The reason for the different colors in the epidermis is, well, they use the differential stain, so kind of like with the pancreas slides, you know, we have the differentially stained pancreas slides that make it very easy to see the islet cells, and the non-differentially stained, where the islet cells are just a little more difficult to pick out. But through differential staining, you can pick out the different layers of the epidermis. So this is that same picture, just a higher magnification. So the stratum basal is that one, maybe two layers, but very deepest layer, very thin of the epidermis. This is the stratum spinosum. This is the granulosum. That light layer is the lucidum. And then that thick layer up here is the corneum. Boys say gorillas like candy. All right, the last thing I want to mention, and we'll go back to the labeled picture just in case you like it. The last thing I want to mention about the stratum basal is there are several distinct types of cells found there. Number one, the keratinocytes, which obvious what they do, they produce keratin. Are you Julie?
Becky.
Becky, okay. Your kindness tried to award a freebie to the class, but nobody knew the answer, so I guess it didn't work out. So we've got the keratinocytes. Another type of cell found in the stratum basal are the melanocytes. The melanocytes produce melanin, as in our pigmentation. We'll come back to that because there's more to pigmentation than melanocytes. There are non-pigmented granular dendrocytes, and these words are all written down for you. You don't have to fight yourself for the spelling. Non-pigmented granular dendrocytes. Those are phagocytes. So the non-pigmented granular dendrocytes would be phagocytizing foreign material that was trying to, or that found its way onto the epidermis. This could be grease because you were working on a car, or some sort of crap you got on yourself working in the lawn, green stain, green dye, whatever. It could be bacteria, it could be dead cells or injured cells, just anything that doesn't need to be there. And then finally, you have the tactile or Merkel cells. The tactile or Merkel cells are receptors for fine touch. Okay, so back, yeah.
What did you say, Merkel cells?
Yeah, Merkel. Back to the pigmentation thing. So the melanocytes and melanin, which are stimulated by the sun, ultraviolet radiation, hence sun tanning. But there's more to skin tone than dark light. We have orange and yellow sort of tones, like a carrot or a squash, and those skin tones come from pigments called carotenes, just like a tree. When the chloroplasts are responsible for the green color that we see in the leaves of a tree, and then this time of year the chloroplasts become less active, and some other organelles called chromoblasts become more active, and they're responsible for the yellow and the orange tones that you see. And of course, if you've ever been to the eastern or southern part of the country, you see many more colors than you see in Colorado. We primarily see the yellow and the gold, and we talk about the aspen changing, and it's beautiful, but we don't get all the brilliant oranges and reds that you see in other parts of the country. Anyway, so our skin has very similar chemicals. The carotenes are responsible for the orangish and yellow skin tones. And then lastly, hemoglobin, okay, as in red blood cells filled with hemoglobin, binds oxygen, transports oxygen in the blood. Hemoglobin is responsible for the pink skin tones. So if you take the dark light aspect from melanin, if you take the yellow, orange, white aspect from carotenes, if you take the pinkish from hemoglobin, a combination of all of that is what determines the skin tone of all of the people that you've ever seen or ever will see. Okay, next page 20. So, Becky, right? You were the spider person. Yeah. Wasn't it a camel cricket? Aren't they like deadly, dangerous? Can't they jump really far? Bite?
I don't know, we'll keep an eye on it.
Anyway, in the interest of your kindness, I tried to give away a test question. It's one that I asked in this room. I cannot believe that nobody was able to answer it. But,
I looked at it.
no, it's not carrot. But I went to, what was it, that one? It was this one. I went to this tissue and I asked what, I mean, you were back in the room when I did it. I didn't want you to miss it. But I asked what protein forms these structures. And I figured somebody would blurt it out. And as a result, this week on the practical, everybody would answer it correctly when I asked it there. Nobody said anything. Nobody knows. The structures are cilia. That's not the question. What's the question? What protein forms these structures? The structures are cilia. What? What? Yes. Thank you. Thank you. Now give us the rest of the details, you brilliant person. They're cilia. They're made of a 9+2 arrangement of microtubules. The microtubules are made of the protein tubulin. The cilia are anchored with basal bodies, which are made of a 9+0 arrangement of microtubules. The 9+2, 9+0 stuff is what? The 9+2 is 9 doublets and 2 inner singlets. That's the cilia and flagella. The basal bodies, the 9+0, what's that all about? It's 9 triplets plus nothing in the middle. Basic biology, guys. Anyway, the answer to the question was tubule. Okay, so I will ask you a question from that world Monday and again on
The
Wednesday.
microbiology teacher would be disappointed.
And we have Becky to thank that everybody gets at least that one question correct because I just gave it away. Of course, most of you would have gotten it anyway. You knew the answer, you just didn't want to blurt it out and sound like a smart aleck. Know it all, right?
Yes.
Mm-hmm. Just humor me and say yes. All right, we're on page 20, Roman numeral 4, the dermis. First of all, if you have your book, get it out, fix it. If you don't have it, make a note to fix it when you get home. These are not layers. They are regions. So there's a papillary region, a reticular region. If you look at your notes, you will see that. In fact, it says dermis, regions, not layers. So the epidermis has 4 or 5 layers. The dermis has 2 regions. So what's the difference between a region and a layer? We'll get there. First of all, layers are simple. Here is a brick wall. A brick wall. How many layers of bricks are in that brick wall? 6. There is nobody in the room or on the planet that can tell me, no, I think that's just 5 layers. Or actually, I think that's 7. It's 6, right? It's definitive. 6 layers. That's the way the epidermis is. A definitive number of layers, 4 or 5, depending on whether the skin is thin or thick. Now, if we have white and black and every shade of gray in between, how many layers would you divide that into?
Infinite.
Infinite. You can say, I want to cut it in half. Somebody else says, well, I want to cut each half in half. I want to cut each half of a half in half. It's like a slice of, like a pie. How many pieces can you cut it into? Infinite. Because you can always cut a piece in half. That's the way the dermis is. At the very top of the dermis, this is your picture again. At the very top of the dermis, the tissue is very, very loose. And deep in the dermis, the tissue is very, very dense. And all you really have to do is compare an area like that to an area like that to see the difference. But it's not that the top fourth or half or whatever is loose and the bottom 3/4 or half is dense. It's a gradual transition from loose to dense. See, we try to force the connective tissues into boxes because the epithelial tissues fit so nicely in boxes. We have simple squamous, we have simple cuboidal, we have simple columnar, we have these highly specific tissues, but connective tissues aren't that way. We force them into their boxes because this is a chapter in the book for us, not a class in histology. But do you really think all you have is loose connective tissue and dense connective tissue? It's kind of like the cloudiness, the sunliness. You know, is it partly cloudy? Is it partly sunny? Is it 50-50? Is it 10-90? Is it 30-70? 70-30? It could be anything, right? What are the degrees between 100% sunshine and so dark and cloudy that we think the earth is ending? How many levels in between those two extremes are there? Infinite. Just like right here. Well, the connective tissues are that way. The fibrous connective tissues. I'm not talking about bone and cartilage, but the reticular, the dense, the elastic, the loose, they are that way. So your dermis does not have any layers. It's got two regions. And even talking about the two regions, a lot of people think is kind of stupid because the number of regions it has is actually infinite. But the point is, the superficial aspect of the dermis is looser than the deeper aspect of the dermis. And whether you want to say, and look at your notes now, whether you want to say the top 25% or the top 30% or the top 35%, it doesn't matter. It's somewhere in there, less than half, versus the bottom 60% or 65 or 70 or 75 is denser. And you may say it's 30-70 and somebody else says, well, I see it as 25-70. Fine, it doesn't matter. Now the numbers I've given you, 20 to 25, 75 to 80, fine. But you could change it to 30-70. It really doesn't matter. All right, some interesting words. I'm looking at letter A, the superficial aspect of the dermis. Where the dermis and the epidermis come together, in other words, this interface, I've said several times now, it's like interlocking fingers, but everything has to have a name. So these dermal projections that stick up are called dermal papillae, and that makes sense. That goes with the papillary region, the superficial aspect of the dermis is the papillary region. So those projections are called dermal papillae, and these words are on your notes, guys. Just look, you don't have to write anything down. And these epidermal projections that stick down are called epidermal parallel pegs. Again, on your notes, just make note of it. Nothing to write down. Huh?
The epidermal parallel pegs.
The epidermal parallel pegs are on page 20, Roman numeral 4, letter A, item 3. So numbers 2 and 3, dermal papillae, epidermal parallel pegs.
I thought it'd be under
Oh. You want to be my next TA? Because I like that idea. You can rewrite the notes. That'd be cool.
I'm a terrible
There's a lot of money in it. I'll triple what I pay Keenan, Shasta, and Molly. All right, Meissner's corpuscles are found in the superficial part of the dermis. I don't have a picture of one, but Meissner's corpuscles are corpuscles. They're deeper than the tactile cells in the epidermis, so therefore deeper types of pressure as opposed to super fine touch. But they're not the deepest type of receptor. We'll get to the deeper receptors here in a minute. Okay, so now we go deeper into the dermis, down toward the, down into the reticular region, the lower 75% or so. And this is where we talk about that hair thing. And remember, I asked you to write down widespread pillow erection. So you'll see hair follicles going deep into the dermis, and every hair in your body, whether it's a hair on your head or a hair on your nose, every hair on your body is anchored in a follicle, a hair follicle. Nobody knows the anatomy of a hair follicle except the people that write the books, and they don't even know it, they just write it. So don't learn it, okay, unless you want to know it for yourself. If I wanted you to know it, I'd have that great big picture out of your book with 50 names on the hair follicle. I could care less about that. Nobody cares about the anatomy of hair and nails. So if you want it, it's in your book, great, but you won't see me ask you about it. What you will see me ask you about are a couple of things associated with a hair follicle. One is this muscle. That muscle is called an erector pili muscle. There are, a couple of others. There's one there. There's a really obvious one right there. You will see this spelled with both an A and an E. Both are correct. Erector pili muscles. They are the muscles that make your hair stand up. We just don't do a very good job of it. For us, we get goosebumps. But what happens if you think about muscles? Muscles run from one point to another. The muscle shortens, and so if this muscle shortens, the bones move. Well, if this muscle shortens, this hair is going to stand up. And so that's what the dog and the cat are doing. So this phenomena of widespread pillow erection is the widespread contraction of your erector pili muscles. Something else that you'll see associated with hair follicles is a type of gland. They're called sebaceous glands, and the words are on your notes. There's the word on the board if you need it. Sebaceous glands, they make oil. Now, nobody knows exactly what the purpose of oil is. As I've said so many times now, who are you going to ask? But we assume the oil is there to moisturize our hair, to keep our hair from drying out, to keep our hair from splitting. Of course, that's almost funny when you think about it. So God or Darwin gave us oil to keep our hair moist and keep it from drying out or splitting. How many billions and billions of dollars does the United States spend each year on products to remove the oil from our hair? What do you think?
A lot.
I mean, I buy Suave shampoo. If I use shampoo at all, when you have hair as short as mine, you generally just wash your hair with a washcloth while you're washing your face. Why use a separate chemical for it? Doesn't matter. But I've seen people buy the expensive shampoos. I know you can buy the really good stuff and all. I have no use for it. But just think about that. Allow yourself to think about that. We have this product that we produce. These sebaceous glands produce it. It's called sebum. And its job is to keep our hair moist. But we spend billions of dollars stripping it out of our hair. And then what do we spend billions of dollars on after we spend the billions to strip the oil out of our hair?
Split-end treatments.
Well, I was going to say conditioner. Don't we? Don't we? I don't. I swear I don't. And if you do, great. A lot of you probably do. But you shampoo your hair, and it's squeaky clean, and then you condition it. Why didn't you just leave it alone? Well, not really. Then we'd all stink. But think about these things. Think about these. And some people really go to the extreme with the let's put the oil back in. Who's heard of hot oil treatment for their hair? This is funny. This is funny. All right, what else do we have here? Well, we have two types of sweat glands. here's the word sweat gland, that's a layperson's word. They're called sudoriferous glands, and you'll see this word on your notes. Let me direct you. Item 6, sweat glands, parentheses, sudoriferous. Yeah,
I was wondering, so the sebum would excrete that?
we excrete sebum because it leaves our body. Yep. Well, if you really want to get tech, we secrete it from the gland into the hair follicle, but it is excreted by virtue of the fact that it's on the hair that protrudes outside the body. All right, so sweat glands, technically sudoriferous glands. The sudoriferous glands are called coiled tubular glands. That's exactly what they look like, a tube that's coiled. The sebaceous glands, the oil glands, are branched, and the word is acinar, which means they're vase-shaped. But if you look at your notes, and I don't have a picture of it right here, I'll find one in a second, but if you look at your notes, you'll see that there are two types of sudoriferous glands. So have a look at your notes there. You see that there are eccrine glands and apocrine glands in the world of sudoriferous. The ones in this picture are eccrine. All right, this is a duct of an eccrine sudoriferous gland. The duct goes to the surface. Let's go back to that last picture. So this is the duct of an eccrine sudoriferous gland. The duct goes to the surface. Remember, the sebaceous glands don't look anything like that. They're not tubular looking. They're not coiled. This other type of sudoriferous gland, an apocrine sudoriferous gland, it's coiled, it's tubular, but look at its duct. Where does it go? To the hair. Yeah, not to the surface, but to the hair. So you've got eccrine and apocrine sudoriferous glands. They're both coiled and tubular. What you want to add about the eccrine versus the apocrine are the following things. The eccrine sudoriferous glands, and this will fit right there on your lecture notes if you want. But, the eccrine sudoriferous glands produce a watery sweat. As in, I am hot and sweating to cool off. Eccrine, watery sweat, hot, sweaty, cooling off. They are present at birth, and they are located all over your body. So you've got four things there. Watery sweat, function is hot, sweaty, cool off. Technical term, as you know, for that is evaporative cooling. Watery sweat, hot, sweaty, cool off, present at birth, located all over your body. The other type of sudoriferous gland, the one where the duct goes into the hair follicle, the apocrine sudoriferous gland, they make an oily sweat. The oily sweat goes with body odor. Doesn't have to be a bad odor. So oily sweat, body odor. They don't become active until puberty. And they're not located all over the body the way the eccrine glands are. They're located in the axillary region, the back of the neck, the genitalia area. so what you should have now, if you look at your notes, when you compare the eccrine to the apocrine, they're both pseudoriferous, they're both coil tubular. The eccrine duct goes to the surface, apocrine duct goes to the hair follicle. Eccrine is watery, apocrine oily. Eccrine, they're all over the body, they're present at birth. Apocrine, they're restricted to like the back of the neck, the underarms, the genitalia area. Hot, sweaty, evaporative cooling versus body odor. The body odor does not have to be bad. Bad body odor, frankly, is more likely the eccrine product, and bacteria are feeding on it, and the byproducts of the bacteria, okay, just like us, we eat, we produce waste. So bacteria feeding on the eccrine sweat produce waste. And that's what's responsible primarily, not only, but primarily for body odor, which is why you bathe and use soap and whatnot. Yeah.
Sorry, is neurocrine just a type of eccrine, or is it like the same word?
It's basically the same word, yeah. You could interchange it. All right, this body odor thing. So there are chemicals called pheromones. And they're similar to hormones. I mean, you've all heard of hormones, but your hormones stay in you. Your hormones don't leave your body. Okay, you don't excrete your hormones. Your hormones don't have any effect on your friend or your neighbor or your enemy. Your pheromones do. Okay, so animals do excrete pheromones, and they are detected by other animals. Remember, you're an animal, so I'm not talking about just giraffes or something ridiculous. The most widely known example of pheromones, this has nothing to do with the integumentary system. Pheromones are in fact found in every body secretion ever studied. The most widely known among lay people example of pheromones, even though they may not know the word, is the male dog marking his territory. If you've ever walked a male dog or seen someone walk a male dog, or a spayed female will do the same thing, but they tinkle, every few feet. And if you're paying attention, why would you be paying attention? Well, because you're weird, okay? You're a scientist. You're curious. That's how you learn. And so if you're looking when the dog does this, sometimes you don't even see anything come out. You see a little drop come out. But he's marking his territory. And again, a spayed female will do the same thing. Other wild animals, deer, elk, they have scents that they release. Some of them go with mating behavior. Some of them go with danger type of signaling other members of the herd. What about people? Well, the two best studied examples of pheromones in humans are, number one, when females live together, sometimes their menstrual cycles become synchronized. This can be a mother and daughter, or it can be a house with a mom and 2 or 3 daughters, or it could be 2 females that live together, share a house or apartment. This can be institutionalized women. This could be the state mental hospital in Pueblo, and every female on a particular unit could be in sync. This is an interesting example of where we ask, why? Why do we have that? Do human females need to have their menstrual cycles synchronized? I can think of no reason why they do. But that takes us to, okay, what about other animals? Do they need to have their menstrual cycles synchronized? Well, in fact, they do. I don't know a lot about non-humans, but I do know that in most non-human animals, especially the things in the wild, not dogs and cats or salamanders that you might keep at home, but animals have a mating season. They have a birthing season. And this revolves around, and we're going to think now about elk and deer, bears in Colorado, Wyoming. They give birth in the spring. Why? Because the babies need the spring, summer, and fall to get big enough to survive the first winter. If an elk gives birth in February, the baby's just going to die. Now, I don't know if they never give birth in February or if they just usually don't. I have no idea. But for the most part, those animals give birth in the spring. So the babies have 9 months roughly to get big enough to deal with the first Rocky Mountain winter. So there is a reason for this synchronization. Why do we have the synchronization? Because God or Darwin, you pick, God, Darwin, some combination of the two, they made us all similar. Mosquitoes, giraffes, human beings, turtles, frogs, lizards, squirrels, we're all similar. We all use ATP as cellular energy. We don't need the synchronization, but we have it. The other widely studied example of pheromones in humans revolves around breastfeeding. They took human females with breastfeeding babies, and they applied every body secretion you can imagine to their breast. So body secretions from their armpits, saliva, yes, urine. I said every body secretion. Pheromones have been identified in every body secretion. When you apply these body secretions to the woman's breast, it encourages the baby to feed. Why do we need that? I don't think we do. Do other animals need that? I suspect they do. How do they know where the food is? And think about dogs and cats. They're born blind, for God's sakes. How do they find the frickin' food? Well, now you know. Pheromones. Why do we have that? Because we're all similar. God, Darwin. That part's personal. You can pick that. But it doesn't matter which one of those you pick. The point is we're all similar. Another not so well studied, but interesting, if I dare use the word, example of pheromones is pheromones are thought to be a sexual attraction. And, you're all adults. You probably cuddled with someone and been attracted to their body odor, not their shampoo or their perfume, but their natural scent. That might be another example of pheromones. Back in the 70s, we attempted to bottle human pheromones. It's called musk oil. I don't know if they make it anymore, but it was popular, I guess, for a while. One summer, probably 10 years ago, I don't know, every time I tell the story I try to add a year to it. Could have been 50 years ago by now, who knows. But it was a summer class, and it was A&P 2, because we reviewed this when we talk about hormones in A&P 2. But after this discussion, some students were out in the rotunda, and one of them had a magazine, I don't know, GQ or Men's Health, something like that. And in the back of the magazine, they were selling pheromones. And 10 or 15 of the students pooled their money. I think they spent around $100. And they received in the mail a bottle about this big that had, well, I don't know what it had in it. I'll tell you what I think it had in it at the end of the story. But anyway, a young man volunteered to be the one to wear the pheromones. And so, we didn't see him, but we all pictured him every morning getting ready and splashing the pheromones on his neck and, coming to school. And for 2 or 3 weeks at the beginning of every lecture, everybody was just dying to hear from what we'll call him 62-7. Anything, dating life, anything to tell, no, nothing. Anyway, nothing in his world ever changed that he shared with us. And after a few weeks, we lost interest in it, and we concluded that they probably paid $100 for a little bottle of donkey urine. Anyway, so the apocrine glands, they are interesting to say the least. That's about it. Let's have a look at the notes. Item 7, Pacinian corpuscles. Let the P in Pacinian stand for pressure. So as you think about, touchy-feely, you've got the Merkel cells for the finest touch, a little deeper, the Meissner's corpuscles, and still deeper, the Pacinian corpuscles, as you think about fine touch versus different degrees of pressure. And there are lots of different types of receptors in there. As I've already mentioned, you have receptors for temperature, receptors for texture. All right, Roman numeral 5, the subq or the hypodermis. You know that's made of adipose connective tissue. Roman numeral 21, or rather page 21, Roman numeral 6, simply summarizes the glands that we've talked about. So the sebaceous glands, the sudoriferous glands times 2, the mammary glands, the ceruminous glands, they come up. The mammary glands, milk, I know you're familiar with the concept of humans and other mammals producing milk to feed their young. But what about the ceruminous glands? Where are those? What are those? Earwax, okay. What's the function of earwax? Anybody know? I mean, it clearly keeps like waxing a car, it helps keep water out of your ear canal. Yeah, it's an insect repellent. It's an insect repellent. I don't know, some of you probably watched some of those different Survivor shows. There's 3 or 4 different ones out there. Some of them are better than others, but you might have seen one where they're in the swamp somewhere being eaten alive by insects and they rub mud all over their bodies and whatever. I'm waiting for the Survivor show where they take earwax. Yeah, let's not share it either. All right, substances produced by the integumentary system, just a list there. I guess you probably want to add melanin. I don't see that on the list. And then finally, Roman numeral 8, epidermal derivatives, recalling that the skin is just an organ. The skin is to the integumentary system as the uterus is to the reproductive system, or the trachea is to the respiratory system. So, what are the other organs of the integumentary system? Well, again, quote unquote, the skin and its derivatives. So what are the derivatives? Well, your hair, your nails, your glands. And if we don't restrict our thinking to people, we could add scales, feathers, hoofs, antlers, but not horns. And I could have that backwards. If somebody could help us out here, please speak up. Antlers are like what a deer has, yes? Okay. Yeah, so antlers are part of the integumentary system. They're derivatives of the skin. Horns are actually part of the skeletal system. Horns are different. So, again, if you want to add scales, feather, fur, hooves. Oh, and I've got antlers written down there, not horns. Okay, good. So it took care of itself. All right, guys, you get out early today. Let's see. Wait. Just so everybody knows, the syllabus talks about this. So I guess you probably know already. But I try after an exam, the first lab after an exam, to hand out your Scantrons and your examination booklets so that you can spend a few minutes and see what you missed and why you missed it, that type of thing. So we'll do that in lab this week, Monday, and again on Wednesday after the practical. So if you want to leave after the practical, you can, but if, you know, if you're interested in looking at your exam, you're free to hang out and do that. And as always, I will grade your practicals, 2-day in the lab, so you, while you're looking at the exam, you could also be waiting for your grade, so you don't have to wait on that. All other grades should be in D2L. That was done over the weekend.
Snake, on the other hand, I'd be on the other side.
I'd be more comfortable with that than a freaking spider, I think. See you later.